Hi, my name is Nuratika Mtiaziz, matrix number 100538.
So hello and hi, my name is Margaret Jamiling and my matrix number is 99626. So in this video, I'm going to talk about the objective of this experiment and also the reduction part of this experiment.
Hi, my name is Muhammad Haikal bin Muhammad Hanif and my matrix number is 99729.
Hi, my name is Khairunisa and my matrix number is 99394.
So what is the objective of this experiment? So the objective of the experiment is to investigate the physical properties of lipid and fatty acid.
Next is material and apparatus. For material, we need microcentrifuge tubes, 6 in total, spatula, dropper and bottles. For apparatus, we need solid, 3 selected lipid-based food, which is olive oil, ghee and shortening, and lastly distilled water.
Prepare 2 sets of 3 microcentrifuge tubes. Add 3 drops of olive oil into 2 test tubes to make 2 sets of olive oil. Add 1 ml distilled water into 1 tube with olive oil. Add 1 ml distilled water into 1 tube with shortening. For the last tube with ghee, add 1 ml distilled water. Repeat the addition using 0.5 ml toluene for the second set of olive oil, ghee, and shortening. Close the lid and shake well. Observe for the appearance, odor, and solubility in water and toluene.
In this practical tree, our analysis focused more on qualitative data. We tested the solubility of olive oil, ghee and shortening in both water and toluene. As expected, none of the lipid samples dissolved in water but all were soluble in toluene, a non-polar solvent. This supported the principle that lipids, being non-polar, are hydrophobic and dissolve best in other non-polar substances. The observations were consistent with the chemical nature of lipid and demonstrated the impact of polarity on solubility.
To conclude this experiment, the experiment showed that ghee and shortening are insoluble in water but dissolve in olive oil, confirming their non-polar hydrophobic nature. This supports their classification as lipids and highlights how solubility is influenced by molecular polarity.
Now we go to the reflection part for this experiment. Firstly, from this experiment we can know whether lipids such as olive oil, ghee and shortening can dissolve in both water and toluene or not. Second one is, this experiment deepened my understanding of how the chemical structure of fatty acid relates to their functions and physical state. This knowledge is particularly important in biochemistry and nutrition science, where lipid properties affects metabolisms, membrane dynamics, and health outcome. For this practical, the tasks were simple but still required focus. We discussed the observations together and shared ideas during the interpretation. It was a smooth and productive group effort. Lastly, I learned about the physical properties of different lipids and fatty acids, such as their melting point, solubility, and texture. It helped me understand how the structure of fatty acid, like chain length and saturations, affects their behavior. Overall, it was a useful hands-on way to connection theory with real example.